So in part 3 of this lecture, we'll talk about nucleic acids. So nucleic acids are polymers that consist of monomers called nucleotides. And the nucleotides itself, they have a recognizable structure composed of 3 parts. They have a 5-carbon or pentose sugar. They have a phosphate group. And they have a nitrogen-containing base that is either a purine or a pyrimidine. So a polymer is synthesized, again, by condensation reaction to make phosphodiester bonds. So like we call the bonds between amino acids peptide bonds. We call the bonds between nucleotides phosphodiester bonds. So there are 2 different nucleic acids. We'll talk about DNA and RNA. So DNA stands for deoxyribonucleic acid. In DNA, the pentose sugar actually isn't a sugar, it's a deoxyribose sugar, and we'll talk about that in just a second. DNA exists as a double helix, so it's double-stranded. There are 2 molecules of DNA that make up the double helix. DNA also consists of 4 different nucleotides that have 4 unique nitrogen bases. Adenine and guanine, those are the purines, and thiamine and cytosine, and those are the pyrimidines. Adenine, hydrogen bonds with thiamine, that is a purine bonded with a pyrimidine. And cytosine, hydrogen bonds with guanine. Again, that's a purine with a pyrimidine. So the order of the nitrogen-containing bases, these form the genetic instructions for every organism because DNA is the genetic information. So this is the structure of a deoxynucleotide. So we have, again, these 3 parts of the individual monomer. We have the phosphate. We have the deoxyribose sugar. Again, this is a 1, 2, 3, 4, 5 carbon sugar, except that 1 of those carbons is missing the hydroxyl end. Right? So it's deoxy right here. And then there is the nitrogen-containing base. In this case, this is adenine, and the nitrogen-containing base is a purine ring. Now, one important concept in these ribose sugar or deoxyribose sugar rings. So we can keep track of the different carbons in this ring chain by numbering them. So we start the numbering starting at that central oxygen and going clockwise. That means this is the first carbon that we count in the ring. So we call that 1' carbon. Here we have the 2' carbon, the 3' carbon, the 4' carbon, and the 5' carbon. And we keep track of things that are bonded or interesting chemical features of each of these carbons, right? Because on the 1' carbon, this is the carbon where the nitrogen base is bonded to. In DNA, the 2' carbon is deoxy, right? It's missing that hydroxyl group. And then the 5' and the 3' carbons, they are important because of the 5' we have this phosphate, and in the 3' we have this hydroxyl. And those form the phosphodiester bonds between nucleotide monomers. So this is the polymer where we have that phosphodiester bond. So again, we have the 5' carbon that's got the phosphate on it. We have the 3' carbon that's got a free hydroxyl. And the phosphodiester bond is formed by a condensation reaction where we have a covalent bond, a covalent ester bond formed between the 3' carbon here, the phosphate, and is bonded to the 5' carbon of the next nucleotide in the chain. Right? So just like chains of amino acids have an N end and a C end, the chains of nucleic acids have an end that has a phosphate group on one end and an end that has a hydroxyl group on the other end. And so this is what we call the 5' end and the 3' end. And it's important to recognize that the 5' end and the 3' end are chemically distinct. Another important chemical property to recognize in nucleic acids is hydrogen bonds and recognizing a hydrogen bond donor and acceptor. Now, hydrogen bonds occur when we have an electronegative atom that has this negative delta covalently bonded to a hydrogen, and because the electronegative atom holds on to an electron, that makes this end of the molecule a little bit more positively charged. Then we have a electronegative atom that has a free pair of electrons, so it's not bonded to a hydrogen. Right? The electronegative atom is able to participate in this hydrogen bond by forming this polar interaction with the hydrogen. So the hydrogen bond donor here is the part of the molecule where we have a covalent bond between the electronegative atom and the hydrogen, and that is going to, I guess what you can think of as donate the hydrogen to the hydrogen bond. And then we have the hydrogen bond acceptor. That's an electronegative atom that doesn't have hydrogen bonded to it, and it can form the hydrogen bond with that hydrogen bond donor, hydrogen bond acceptor. So let's look at the individual bases that make up DNA and look at where we can find some hydrogen bond donors and hydrogen bond acceptors. Here on the ring of adenosine, we have an electronegative nitrogen that has hydrogens bonded to it. So here we have a hydrogen bond donor. Here we have a nitrogen that's covalently bonded to carbons and is missing a hydrogen on it. And so that is a hydrogen bond acceptor. Again, on cytidine, we have a hydrogen bond donor, acceptor, and acceptor. Whereas on guanosine, we have a hydrogen bond donor, donor, and acceptor. We look carefully at the structures of each one of these individual nitrogen bases and make sure you understand where what functional groups are donors and what functional groups are acceptors. As this pattern of hydrogen bond donors and acceptors on each one of these nitrogen bases allows these nitrogen bases to form complementary hydrogen bonds and fit together almost like puzzle pieces. Right? So if you look carefully at the structure of thymine, right, we have a oxygen that is missing its hydrogen attached to it. Right? Whereas on adenine we have an amine that has these two hydrogens to donate. And so hydrogen bond acceptor, hydrogen bond donor form a complementary hydrogen bond. Here we also have another hydrogen bond donor, hydrogen bond acceptor. And so these two in the geometry of thymine and adenine form these puzzle piece interactions because they have these two complementary hydrogen bonds right on the right places in the molecule. Similarly, guanine and cytosine fit together just right because there's a hydrogen bond acceptor, a hydrogen bond donor, hydrogen bond donor and a hydrogen bond acceptor, and a hydrogen bond donor and a hydrogen bond acceptor. And so they can only form these complementary interactions in one way. So these puzzle pieces only come together when the two strands of DNA that are joined together by phosphodiester bonds, shown here, line up in just the right way in what we call anti-parallel. So the 5' end of one chain is opposite of the 5' end of the other. So let's take a look at that overall three-dimensional structure of DNA. We represent the structure of DNA in a number of different forms, just like we represent the structure of all complex organic molecules. So we have this cartoon version of DNA over here on the left, where we see the double helix wrapping around each other. We have these nitrogen bases on the middle that form these complementary hydrogen bonds. Around the outside we have these covalent linkages in the individual molecules that are formed by those phosphodiester bonds. So we have the phosphates and sugar groups on the outside end. And then those form these complementary interactions to form the double helix structure. Right? So if we look at the double helix, we have these nitrogen bases again on the inside of the double helix, forming these complementary hydrogen bonding interactions. 2 for adenine and thymine, and 3 for cytosine and guanine. And then we have these chains of phosphate and deoxyribose sugars on the outside. Now these complementary hydrogen bonds between A's and T's and C's and G's can only form that right geometry if the 5' phosphate of one end of the strand is opposite to the 3' end of the other. Right? So one of those chains in the double helix is running 5' and 3' this direction. And the other one is running 5' to 3' in the opposite direction. So they have these complementary base pairs, but they're also anti-parallel to each other. So this concept becomes super important when we represent DNA, the primary structure of DNA, as a sequence of those 4 DNA bases. Right? So we can write the primary structure of DNA as a sequence of bases. So the sequence of bases, DNA doesn't have a left and a right. It's got an end with a phosphate on it and an end with a free hydroxyl. That's the 5' phosphate and the 3' free hydroxyl. And so we can write the nucleotide bases starting at, for example, the 5' end, and then writing them until we get to the 3' end. So if we look at this monomer, or this DNA up here, and look at the monomer units, the 5' end, the 5' phosphate of this monomer is over here, and the nitrogen base that's attached to it is a C. And so the structure that we'd write is 5' C. The next nucleotide on the chain is a G, and so that's here. Then followed by a T, and then an A when we get to the 3' end. If we look at the complementary molecule down here, we have the 5' end of the molecule, and the first nucleotide is a T, then an A, then a C, then a G. So that's 5' T, A, C, G. Right? So these complementary interactions only occur when the 5' end is opposite to the 3' end, and we have these complementary bases. Right? So the sequence of this complementary molecule, so these are shown with these complementary hydrogen bonds, is 5' C, G, T, A, and this one is 5' T, A, C, G. Right? So when you're writing complementary molecules, you need to make sure that those molecules are complementary and anti-parallel. So again, A pairs with T and C pairs with G, but also needs to be anti-parallel. Right. So if we were writing the sequence of this molecule up here, we would write 5' C, G, T, A. But the complementary molecule to that, if we wrote the complementary bases, we have G, C, A, and T. We know that G pairs with C and A pairs with T. But we also need to remember that the 5' and the 3' need to be opposite to each other because these are anti-parallel. So this molecule here, 3' G, C, A, T, is complementary to this molecule here. Right? This 3' G, C, A, T, right, is just the sequence of this molecule written from the 5' end, and then ending at the 3' end, or starting at the 3' end and ending at the 5' end. But if we were instead write the sequence of nucleotides in this chain, starting from the 5' end and ending at the 3' end, that sequence would be 5 T, A, C, G. Right? These are the same thing. We're just indicating which end we're starting writing from, the 3' end or the 5' end. So keep this in mind as we practice writing the sequence of these complementary molecules in class. So the other nucleic acid is RNA. RNA has a couple of many similarities to DNA, but a couple important differences. So one, in DNA, remember we have this ribose sugar that is actually missing, it's not a sugar, it's missing an oxygen right here. But RNA is ribonucleic acid. It has a full sugar, it's got the hydroxyl on that 2' end. The other important difference between RNA and DNA is a very minor one. It's the identity of the nitrogen base. Right? So thiamine is a nitrogen base found in DNA, and uracil is a nitrogen base found in RNA. Right? So thiamine has a methyl group on the nitrogen ring, whereas uracil does not. But if we look at the hydrogen bond donors and acceptors in thiamine and uracil, those are exactly the same. And so the only difference is not actually involved in the base pairing of the DNA or RNA. So in DNA, thymine pairs with A. Right? In RNA, we have a uracil that also pairs with A in the exact same way. So uracil in RNA can pair with adenosine in RNA and also adenosine in DNA. Thymine in DNA can pair with adenosine in DNA and also adenosine in RNA. So two important differences. Ribose nucleic acid contains uracil. So these two properties of RNA create a little bit different behavior of RNA inside of cells. Right? So in RNA we have G that pairs with C and A that pairs with U. There's no T. The other important thing is that most RNA is not formed as a double helix. It's single-stranded. So there's just one molecule of RNA. And there's no complementary strand made. But RNA can still form complementary hydrogen bonds, and so often those bonds are with itself. They're bonds within the same strand of molecules. Right? This allows RNA to behave a little bit like a protein, where RNA is able to fold into complex and diverse three-dimensional structures, and it gives RNA a number of important functions in the cell. So in viruses, remember in the beginning of the class we said viruses can have genomes, unlike cells that have just DNA genomes. They can have RNA or DNA genomes. And the RNA genomes of many RNA viruses have interesting structures that contribute to their viral replication, such as the three-dimensional structure shown here of the Zika virus genomic RNA. So these three-dimensional structures are formed by RNA folding and making complementary interactions within the molecule. Right? So here we have a chain of RNA bases. And if you look very, very closely, you will see that this sequence of RNA bases here is complementary to another sequence of RNA bases found later in the molecule. Right? So if that RNA folds on itself, these strands of complementary bases can base pair with each other because the 5' end of this side of the hairpin, right, is opposite to the 3' end of the other side of the hairpin. And all of these bases are complementary. Right? So this is a structure, again, that we call an RNA hairpin because we have a complementary hydrogen bonding interaction between this stem, and then we have the turn on the end. So we call this either a hairpin or a stem loop structure in RNA. So as I said, the complex structure of RNA allows it to have many different functions and roles that include messenger RNA. So messenger RNAs are single-stranded RNA that carry the genetic information from DNA to the ribosome. So the ribosome is a complex inside cell that carries out the making of proteins. Ribosomes are made of both proteins and also ribosomal RNA, where there are long, complex, three-dimensional RNAs that form the large structure in the ribosome. An important fact about the RNA in the ribosome is that the RNA in ribosomes actually serves as an enzyme. Right? It catalyzes the formation of the peptide bonds in amino acids. tRNAs also participate in translation. So they are three-dimensional RNAs that match each codon in the mRNA with an amino acid during translation. We also, again, have viral genomic RNAs, right? And we have many RNAs that perform lots of interesting functions in the cell, including regulating genes like microRNAs and long non-coding RNAs.